velocities at lower concentrations of substrate. And then when you throw in the inhibitor, or negative effector, then you see it takes more substrate to get it up to the same velocity as without inhibitor or with the activator instead. So ATP increases activity and CTP decreases activity. And since C is a pyrimidine, this is an example of feedback inhibition as well. So in this case, you've got some pathway, A going to D, and D can come back and can inhibit that earlier step, and that's called feedback inhibition. So just to be clear, not every single case of allosteric regulation is feedback inhibition, but in this particular example, it is. So since CTP is a downstream product, this is also an example of feedback inhibition. Covalent modification. We had our different types. The most common is phosphorylation. If you have a protein the enzyme that phosphorylates it is called a kinase. And ATP comes in, ADP leaves, and you end up with a protein that is phosphorylated. If you have a phosphorylated protein, and a phosphatase, and you bring in some water, then you end up with your protein that is now dephosphorylated plus an inorganic phosphate. What class of enzyme is a kinase? How do you know? Yeah, what am I transferring? Phosphate group, yeah.
Is that water molecule leaving?
No, coming in.
It's coming in. Oh.
It's coming in. And my phosphatase, a class. Hydrolase. How do I know? Water, okay. And I'm breaking a bond with it. So I'm hydrolyzing, this will be a phosphoester linkage for the phosphate. So I'm hydrolyzing off the phosphate. And I know that in the homework there was this question that stumped a few of you. These are not reverse reactions of each other. So they have opposing effects in that I have a protein that is phosphorylated or not phosphorylated, but the reaction itself is not the reverse reaction. Yep.
Why is it not a ligase?
Why is it not a ligase? Ligases take ATP. They consume ATP.
Well, aren't you doing that?
but it's not the phosphate that is being joined. So you can't, so when you say that you consume ATP, it's to join two other things
Oh,
together,
okay.
not, yeah. So here, yeah, we're using up ATP, but it's not to join two other things together, it's just moving phosphate through it.
Thank you.
Okay. An example is glycogen phosphorylase. Which is regulated by opposing kinases and phosphatases. So this is actually the enzyme that you need in order to break off glucose from glycogen at the non-reducing ends of glycogen. So when we talked about, you want to mobilize your energy really fast, you want to cut out all those non-reducing ends because you've got thousands of them compared to this one reducing end. And so the enzyme that does that is glycogen phosphorylase, and it's called a phosphorylase because it's actually breaking a bond not with water but with phosphate instead. So it's a kind of a different reaction there. You get a glucose 1-phosphate, and this activity is regulated by kinases and phosphatases, and I've only shown you one teeny little piece of a very large kinase phosphatase signaling pathway. So it's just kind of the last step of this pathway. But glycogen phosphorylase has two forms. It has this B form and this A form, and depending on its phosphorylation state, that determines which form is more likely to be in. So the B form is not very active. The A form is active, and it will adopt the A form if it's phosphorylated. So in this case, we see increases in activity with phosphorylation. That is not always the case. Sometimes an enzyme is negatively regulated by phosphorylation, so you can't assume that phosphorylation is always active. So there is a kinase called phosphorylase kinase that phosphorylates glycogen phosphorylase, and then there's the opposing phosphoprotein phosphatase 1, which breaks off the phosphate group and turns it back into the B form. The names are not very exciting, and they seem to follow kind of a path. So I'm phosphorylating a phosphorylase. But phosphoprotein phosphatase 1 is this random name because it cuts phosphates off of everything, pretty much everything in it matters. Questions? Lovely. You ready to actually do some math today? Hopefully you have a calculator. Okay, activity.